I'm a CLL patient advocate in Melbourne, Australia, and I'm really sorry I can't be with you this weekend, but I'm very grateful that I've still got the opportunity to speak to you and tell you my story. I was diagnosed with CLL at the age of 38, a week before Christmas in December 2011, and at the time I was told that my markers suggested that I would have a life expectancy of somewhere between 5 to 7 years. I had my 2-year-old daughter on my lap on the day I was diagnosed, and as devastating as the diagnosis was, I was determined that there would be something out there that I would hopefully find in time to save my life. Now, in 2011, for a 38-year-old patient, the standard treatment was watch and wait, followed by FCR chemotherapy, and at that time, the doctors really wanted to put off doing chemo for as long as possible, because once chemo failed, we would have to go straight to transplant. And for those who aren't aware of the risks of transplant, there's some research suggesting that mortality rates can be as high as 40% in CLL from stem cell transplants. So it was obviously something I wanted to put off for as long as possible. At the time I was diagnosed, I had 3 children. They were 7, 5 and 2. I was put into watch and wait, and at that time I started looking at international forums, started reading medical journals. I'm a journalist by trade, so really tried to arm myself with as much information so that really I had choices and could, establish what the best treatment would be to give me the longest life possible. And I even had things such as very early on I decided that it wasn't quality of life that was important to me, it was quantity of life. So if there was something that I would have to take or do to extend my life that affected my quality of life, as long as I was there to see my children reach adulthood, I would do that. In 2012, so a few months after diagnosis, I started getting very sick. My lymph nodes were growing. I have the SLL form of CLL, so it's far more, the cancer grows in my lymphatic system, and my neck ended up bigger than my head in the end. And fatigue, losing weight without trying, just all picking up every infection that my children brought home. I got pneumonia. I got sick very, very quickly, and at that point the doctors were pretty much insisting that I had chemotherapy. But mid-2012 there was PCI 37625 in trials, and that has subsequently become ibrutinib, and I really wanted to try and see if I could find a frontline treatment of a novel therapy rather than having chemo, and put off having chemo for as long as I could. Unfortunately, in Australia in 2012, there was no way to access ibrutinib, and by the end of 2012, I was approaching another Christmas, and my doctors said, you have to have FCR, and we'll let you get through Christmas, but we'll start the first week in January. And I was very sick over Christmas, got to January, had the first cycle of FCR, and immediately my neck went down. I could turn my head. I felt $1 million. And I remember turning around to my doctor, one of the doctors who'd insisted on the FCR, and said, why didn't you tell me I was going to feel this good? I would have done this months ago. And she sort of rolled her eyes. So I've always been very determined when I'm telling my story to stress that FCR actually saved my life. As much as I didn't want to have chemotherapy, there is still a role for it in this disease. While there are all these wonderful drugs out there, some people, if they have the right markers, there's statistics that if you're mutated with del13Q in CLL, 40% of people who have FCR have not relapsed. And we're talking about people now who are 15, 20 years out, and that's effectively they're cured. For me, unfortunately, going into FCR, we knew I didn't have the right markers. I was unmutated. My only sort of bad marker is deletion 6q, which isn't a particularly well-known one in CLL, but it's very well-known in lymphoma. It's seen as an intermediate marker in CLL. and it's not normally tested for in fish. That's why most people wouldn't find it, but in Australia they did test for it and they found it. The other thing at the time in 2012 that they didn't test for in Australia was the IVGH test, and that, for those who don't know, you're either mutated or unmutated, and if you're unmutated, you're much more likely to have aggressive disease, relapse early, and the disease is highly resistant to treatment. And I paid for that test to be done privately here in Australia because I wanted to know. And the theory, I think, why they didn't offer that as a standard test in Australia at the time was it actually made no difference to the treatment you got here. We only had FCR as a choice back then. Even if you were 17p unmutated, you would have still had FCR in Australia in 2012. Anyway, I had 3 cycles of FCR. We found out I was MRD negative in my blood, and that was enough to end treatment there and really give me another period of watch and wait. They were hoping I would get a remission of somewhere 2 to 5 years. 5 years would have been fantastic, and then the next treatment option was going to be a stem cell transplant. Unfortunately for me, I don't have a sibling match. And I do have an unrelated donor in Germany, and there's a couple of good matches in Germany. So I've had, consults with a transplanter every 3 months for the whole of this journey because transplant was very much the next treatment I was going to have. 6 months after chemo finished, we started seeing CLL back in the blood. And that was pretty devastating that I'd only really got 6 months of remission before the disease was taking off again. And it took off at a steady rate, but enough to require treatment. And I was getting lumpy again, sick again, tired, the night sweats. And these are drenching night sweats where, those of you who have this disease would know you're not just, a little bit clammy of a night, you're actually changing your entire bedding because you're so sick. And I got to mid to late 2014 when we were preparing me for transplant, and the plan was to give me another couple of cycles of FCR and then we would do a full allogeneic transplant. But by this stage, some of the research that was coming out about survival rates with unrelated donor transplants were really starting to scare me. I was only 40, the children were only 9, 7 and 5, and my biggest concern, or 4, Tasha was only 4, my biggest concern was that I wanted particularly Natasha to remember me. So if I was going to have to do something as serious as transplant, I really wanted to do it later in the disease rather than at this stage. I still felt well enough to be a mum. I was still working, but I was very scared of transplant, and most of you in the room would appreciate that. So after agreeing to sort of go down the transplant route, I asked my doctors if I could do one last-ditch thing and go to America for a second opinion and to see if I could find a clinical trial. There was a very famous Australian called Ron Walker who'd actually joined a clinical trial in California for melanoma, and he really inspired me to do the same. So the doctors here were fantastic. They sent me, they allowed me to go, and I saw Thomas Kipps at UCSD, who was amazing. I had just emailed him and he said, yep, sure, if you come, I'll see you. And they made an appointment for a couple of weeks later, and I had a 2-hour consult with Thomas Kipps, and his main headline for me was, do not have a transplant, we have some drugs. And ironically, one of the drugs, one of the compounds that's in development is called ABT199, and that's actually been in trials in your hometown of Melbourne. And this was actually the first time I'd heard of this drug, and I'm sitting in a hospital in California. And he said, but we can't put you on a trial here. If you have a heart attack on a phase 1 clinical trial of a new drug in America, you will end up with $1 million, or your estate will end up with $1 million bill. But while you're here, come to a conference we've got on tomorrow. It is about clinical trials, and hopefully you'll be able to get some information there that you can take back to Australia and see what's available to you there. So I went to the conference the next day, and a Scottish doctor was talking, and I turned to the patient next to me in this room of 200 people, and I know Brian Kaufman will be in the audience here. Brian organised this conference for the CLL Society, and I'll be forever grateful for that, because I really believe this meeting saved my life. John Gribben from Bart's Hospital in London finished talking, and he came and sat next to me, and he turned to me and he said, "Oh, you've come a long way," because I had a little koala on my jacket, and I said, "Oh yeah." He said, "Are you a carer?" And I said, "Oh no, I'm a patient." And he said, "Wow, how old were you when you were diagnosed?" And I said, 38. And he said, "But you haven't had treatment." And I said, "Yes, unfortunately I've failed chemotherapy and I'm heading for transplant." And he said, "Have you still got your British passport?" I said, "Yes." And he said, "If you can move back to England in the next couple of months, I can get you on a trial of Obinutuzimab and Venetoclax. It's a phase 1 trial. It's a phase 1 combination trial. Both drugs have been in phase 3 trials, so they've been established as being safe. They haven't been put together, but there's no reason why to think that is not a good combination. And if you take the risk, I'll get you on the trial." And so I came back to Australia. I saw my doctors here, and they actually looked at this trial and said, "You've got to do this. It's your best alternative to transplant, and if it doesn't work, it will be the reason why you go straight to transplant." And later one of my doctors here admitted that they were really seeing, actually I think it was John Gribben admitted, they were really seeing this trial as a bridge to transplant for me. So the intention was still to transplant me, but to get the disease under control by me entering this trial. Moved back to England, and within a couple of months I was patient 49 on the trial of Obinutuzimab and Venetoclax. Now this trial immediately came after the sad deaths of 2 patients in America from tumor lysis syndrome on the monotherapy trial of Venetoclax, which I know you'll all be very aware of, and that nearly brought an end to Venetoclax at that point. They just didn't appreciate how powerful a drug this was. So when I took my 20 milligrams of Venetoclax for the first time, I was terrified. I was sitting in a hospital bed, dialysis machine quite close to me, and John Gribben saying, "Don't worry, Deborah, we're following the Peter Mac protocol," which ironically is my hospital in Melbourne, because they'd had 80 patients on the monotherapy trial here and hadn't lost anyone to tumor lysis syndrome. So the protocol was changed, and we had a very, very slow dose escalation to 400 milligrams over 5 weeks. So this was October 2015. I was on the full drug by January. I had Obinutuzimab, 6 cycles of it, 1 day a month, and they used it. I had, I think, 2 cycles of it before we even introduced Venetoclax to lower the tumour burden. The impact was immediate. I went down from, I had 81% infiltration of CLL in my blood, and it was gone within a matter of weeks. It took, I think, 4 months technically to have complete remission. And then by the following December, so December 2016, I was told that I was MRD negative with no detectable disease in my bone marrow. And I'll never forget that, because that was the first Christmas I'd had good news in 5 years. So it was an incredible journey. It did involve me moving to London for 7 months. I left my children in Australia. They visited me, but this was something I had to do if I was going to be there for them for the long term. And even once I finished the initial 7 months on the trial, I still had to be in London 1 day a month. So I commuted for a further 10 months, and then I commuted every 3 months from Melbourne to London. So it was quite the journey, but completely worth it. So we're now coming up to 3 years since the trial began. I'm still in complete remission. There are some technical debates about MRD negative, MRD positive. I've been having 500,000 blood cells examined through the flow cytometry system for 3 years, and now all of a sudden they're looking at 4.5 million cells. So There is detectable CLL there, but depending on where you're on the assay, I'm either just MRD positive or I'm still MRD negative, but I will take complete remission any day of the week. I stay on 400 milligrams of Venetoclax. I don't have any side effects. The only side effects of Venetoclax I had through the whole journey was a bit of flushing every time I dose escalated. And I did pause the drug for 4 weeks last year because I got food poisoning and I just couldn't clear it. And they wanted to make sure that the gastric upset wasn't caused by Venetoclax. They thought it was unlikely, but it is known, so they got me to pause the drug. And then when I restarted the drug 4 weeks later, and it was established it didn't cause the gastric upset, when I re-established it, I went straight back onto the 400 milligrams and immediately got the flushing. So I should have probably dose escalated back on, but you know, I'm still probably one of the longest people to be on the drug. So we're learning. I feel well. I'm working full time doing this in my office in Melbourne. And just very grateful to be here. And I really think my job now as a patient advocate is to tell my story to give patients hope, because when I was diagnosed in 2011 there was no hope. I was told I was going to die, and it even, you know, it took 4 months before I even heard about bone marrow transplants and the fact that this could save me. And if I relapse or become resistant to Venetoclax, I may still be looking at transplant. So I'm very conscious that I need to keep ahead of the science, or at least on track with the science. I'm still reading papers, I'm still taking part in forums, and as you all know as patient advocates, the fact that we're learning from each other and helping each other, and you know, I've got wonderful friends in this community, and you know, I know Michael from Ireland, Michael Rinn will be there, and I'm just so grateful that we're sharing this journey together, and I'm so grateful to the scientists for giving us the hope that we have. Sorry I'm not there, but I really hope I'll be there to see you all and enjoy the conference next year. Thank you.